In the last period, my dear students, we have studied about the glucose. Glucose is the substance that occurs in a great amount in the natures. We can say that it is found in free in natures, and it is found mostly into the fruits and honeys, and the sweet substances like ripened grapes have also huge amount of glucose with them. The various methods of preparation of glucose can be prepared from cane sugar. You can see that sucrose in the presence of water and in the presence of sulfuric acid or HCl or in the alcoholic medium, we get glucose and fructose. Remember that glucose and fructose have the same kind of molecular formula. You can even prepare glucose from the starch. So for preparation of glucose from the starch, we take starch or cellulose and then allow it to undergo the process of hydrolysis in the presence of dilute sulfuric acid at 393 Kelvin temperature and under high pressure, that is almost 2 to 3 atmospheric pressure. We get huge amount of glucose by this process. Glucose is also known as the dextrose, and the molecular formula of glucose is C6H12O6. Same way, my dear students, glucose is an aldohexose because aldehyde function group is present. Glucose is also known as dextrose. Glucose is the monomer of many uh, uh, many larger carbohydrates like starch and cellulose, and that is why we can prepare glucose from starch and cellulose. Glucose is probably the most abundant organic compound on the earth, and the structure of glucose is derived from the various chemical reactions. So now let us study the various chemical reactions on the basis of what we can derive its structures. Glucose, when it is allowed to heat, or glucose when it is heated prolongedly with hydrogen iodide, it gives the normal hexane, which says that all the carbon present in glucose have the linear structures, or they are linearly arranged. Glucose when reacts with hydroxylamine, it gives oxide. Glucose when reacts with the HCl, it produces cyanohydrine. And from these reactions, we can say that glucose has got the carbonyl group present into it. Glucose, my dear students, when it reacts with the bromine water, which is the mild oxidizing agent. So glucose, when it is mild oxidized with the bromine water, glucose produces the gluconic acid. This gluconic acid contains uh, COOH group on the first carbon, whereas the CH2OH group on the last carbon. From this, we can say that carbonyl group is present on glucose as an aldehyde, because the aldehyde group can be oxidized to carboxylic acid by mild oxidizing agent, but this alcohol group has not been oxidized. When glucose is oxidized and with the strong or vigorous oxidizing agent HNO3, glucose produces a substance which is known as the saccharic acid. This saccharic acid has got COOH group on the first carbon as well as the COOH group on the last carbon. That we have even seen that when glucose undergoes the mild oxidation, there is a COOH group on first carbon and CH2OH group on the last carbon. This gluconic acid, which is produced from glucose by the mild oxidation, when it is subjected to the vigorous oxidation, it even gives the saccharic acid. That means this CH2OH group is even converted into the carboxylic acid group, which was not converted to carboxylic acid by the mild oxidation. So we can easily realize that on the first carbon we have CHO or aldehyde group, but on the last carbon we have CH2OH group, and that is why we can say that primary alcoholic group is present in glucose on the last carbon. Same way, my dear students, on the when glucose is subjected to the acetylation process. In the process of acetylation, we use acetic anhydride for the acetylation. So glucose, when it undergoes the process of acetylation, 1 mole of glucose requires 5 moles of acetic anhydride and produce glucose pentaacetate. That means 5 acetic anhydride are being utilized for the acetylation, and that is why we can say that in glucose we have 5 OH group present. Now, if there is 2 or more than 2 OH group present on single carbon, it is not stable, but glucose is stable. That means there must not be two OH group present on the same carbon. And on first carbon, glucose had CHO. That is why all the 5 other carbons have single OH group present in the glucose. And from this reaction, we can conclude that glucose has 5 OH group present. All 5 OH groups are bonded to the different carbon, because two OH group on the same carbon is not stable, and glucose itself is a stable compound. And on the basis of that, we can easily understand the structure of a glucose. Now, on the basis of all these reactions, we can find out that glucose has a structure in which the first carbon has got the aldehyde group, second, third, fourth, fifth, and sixth carbon. Sixth carbon has got a primary alcohol CH2OH group. On the second carbon, on the fourth carbon, and on the fifth carbon, these OH group present are on the same side, but on the third carbon, the OH group present is present on the opposite side. There is a particular way by which we can decide the capital D and capital L for glucose. We have studied into the chapter haloalkanes and haloarenes that if the substance rotate the light or of plane polarized light into the right hand side, we call it the plus glucose or plus sign we apply it. And if it is or we can call it as a dextro-rotatory, and if it rotates in the anticlockwise, we call it levo-rotatory or minus. Thus, plus glucose and minus glucose, we understand by its ability to rotate the light into the clockwise and anticlockwise manner. But we also call D, capital D glucose, and this capital D or capital L is given on the basis of the structure of the glyceraldehyde. So to understand this, my dear students, we need to understand the various steps of doing it. So to understand the various steps, my dear students, let me explain you how we can determine the capital D or capital N for glucose. For doing that, first we need to find out first we need to arrange all the carbon present into the substance. So here in glucose, we are well aware that all the carbons present into the glucose, they are in the linear form. We have 1, 2, 3, 4, 5, 6 carbon. We are already aware that on the first carbon we have COOH group, and all the others have OH or alcoholic OH group are present. And on the sixth carbon we have CH2OH primary alcohol group. The OH present on the third carbon is on the opposite side. So this is how we understand the structure of the glucose. Now we have arranged all the carbons into the glucose. Now on the basis of the priority, we just need to give the numbering to the all the carbons. So as aldehyde is the functional group, we give it as a number 1. This is number 2 carbon, this is number 3, this is number 4, this is number 5, this is number 6. Now we need to find out the carbons which are optically active or chiral carbons. So you can see all these carbons present in the glucose are chiral carbon. Now in the next step, my dear students, we should know that here this sixth carbon has 2 hydrogen present on it, and that is why this is not chiral carbon. Now we need to find out the chiral carbon which has got the highest number. So the fifth carbon has got the highest number. You can see on the fifth carbon the OH group is present on the right hand side. So for determining the capital D or capital L, first we arrange all the carbon, then we give them the number on the basis of the priority for the functional group. Then we find out all the chiral carbons, and the chiral carbon which has got the highest number, for that carbon we look about the OH group present on which side, my dear students. So here in glucose we can see that the OH group is present on the right hand side, my dear students, and on the basis of that, my dear students, we will understand about the glucose. So now we compare the structure of glucose with D-glyceraldehyde and L-glyceraldehyde. You can see that in D-glyceraldehyde, the OH is present on the right hand side, and in L-glyceride the OH is present on the left hand side. So on the basis of that we can call it as a plus capital D or capital L. Here in glucose also we have seen that on the fifth carbon the OH group was present on the right hand side, and that is why we apply the capital D before the name of the glucose, and that is why we call this glucose as a D plus glucose. So this is how we give the name capital D before the name of the glucose. Apart from the capital D plus glucose, we even get capital D minus glucose, capital L plus glucose, and capital L minus glucose forms are also formed. Now, my dear students, we have come to know about the structure of the glucose, but glucose does not give some of the important tastes, like glucose does not give the 2,4-DNP taste, glucose does not give the stiff base taste, glucose does not give the hydrogen sulfide taste. As glucose gives some of the taste, like the taste with HCl, it gives cyanohydrin. When it is allowed to react with the NH2OH, that is hydroxylamine, it gives oxide. But these tastes are not given by glucose. Then there must be something that we need to even study more about glucose. So when we crystallizes this glucose, my dear students, we came to know few important things about glucose. While we were crystallizing the glucose, we came to know that crystallization when crystallization is done for glucose in the aqueous solution of glucose at 303 Kelvin, for the concentrated solution, glucose is formed with the melting point of 419 Kelvin. Same way, my dear students, when the crystallization of glucose is done from the saturated solution, which was hot saturated solution, at 371 Kelvin, we get a glucose which is having the boiling point 423 Kelvin. So we got two different type of glucose which are having even their melting point different. These two crystalline forms shows that they might have different structures. So glucose is existing in a two different structures which have two different melting point, and they need to be different in their structures. So now we have two different information. Sorry, dear students, that glucose does not give few taste of aldehydes, and we conclude that glucose does not have free carbonyl compound. This carbonyl group present in glucose is not always free. That means it must be sometime in the combined state. At the same time, when crystallization is done, aqueous solution of glucose in concentrated solution at 303 Kelvin give glucose with melting point 419 Kelvin, whereas in hot concentrated solution at 371 Kelvin, we get glucose that is having the melting point of 423 Kelvin. These two forms of glucose are called the anomers of glucose. Please remember, during the crystallization of glucose, we get two forms of glucose. One is having the melting point 419 Kelvin, one is having the melting point 423 Kelvin. These two forms of glucose are known as the anomers of glucose, and they are called alpha D plus glucose and beta D plus glucose. So these anomers of glucose are called the alpha D plus glucose and beta D plus glucose, or alpha form of glucose and beta form of glucose. Now let us understand the structure of glucose in detail. Suppose we have glucose. When it is allowed to react with the 2,4-phenyl hydrogen, it gives us these structures. So it combines with the phenyl hydrogen and gives us the salt. Then you can see that this carbon oxygen bond and this double bond undergoes the rearrangement, and finally we get this oxygen and carbon has got the double bond, and this C6H5 and N into NH has been changed, and we get the carbonyl group on the second carbon, which in turns out to be the another form of glucose. You can see that CH double bond and NHC6H5. See again, if this form, when we allow the glucose to react with the phenyl hydrogen, it gives us this product. where we have the carbonyl group present on the second carbon. When the another molecule of the phenyl hydrogen reacts with the glucose, the another molecule of phenyl hydrogen is bonded to the second carbon, and this reaction with phenyl hydrogen gives us the oxazone as a product. This, this same kind of product, now, this same kind of oxygen is also obtained from the D-mannose. In this D-mannose, if you look, it has got the similar kind of the structure of glucose. Once again, my dear students, let us understand this reaction in a very different manner, or once again understanding when glucose is allowed to react with the phenyl hydrogen, it gives the glucose phenyl hydrazone. This glucose phenyl hydrazone by the rearrangement get the carbonyl group present on the second carbon. When this another glucose phenyl hydrazone, when undergoes the reaction with the phenyl hydrazine, it gives a new compound which is oxazone. The oxazone formed from the glucose has same structure as the another sugar D-mannose gives with the reaction. When this D-mannose is allowed to react with the phenyl hydrazine, it gives the same kind of products as it is obtained or as it is obtained from the two times reactions of glucose with the phenyl hydrazine. Please remember, when we do the reactions of phenyl hydrazine with the glucose, it consumes the two molecules of phenyl hydrazine and gives us the oxazone. The oxazone that we get from glucose is very similar when we allow this D-mannose to react with the phenyl hydrazine. Now let us, the same oxazone given by the another substance called D-mannose. So D-mannose and D-glucose are called the epimers of each other. Please remember, my dear students, that this D-mannose and D-glucose are called the epimers of each other, whereas the alpha D plus glucose and beta D plus glucose, they are called the monomers of each other. So many a time, if you may be asked about the epimers and anomers. So D-mannose and D-glucose are called the epimers, whereas the alpha D glucose and beta D glucose are called the anomers of glucose. Note, my dear students, that D-mannose and D-glucose are called the epimers of each other, and alpha D glucose and beta D glucose are called the anomers of each other. Now, what happens, my dear students, that as the open structure of glucose does not give few tests, it is believed that the glucose has a cyclic structures also. So we have seen that some of the tests of carbonyl group is given by glucose and some of the test of carbonyl group is not given by glucose. So glucose may have the open chain structure as well as glucose may have the cyclic structures. So glucose has two structures. Two structures is believed to be obtained by the reaction of aldehyde group on C1 and OH group of C5. You can see that. Let us look at the open chain structure of glucose. This is the open chain structure of glucose. This is carbon 1, 2, 3, 4, 5 and 6. This C double bond O group present on the first carbon and the fifth carbon that has C bond O group. Here the reaction between these two carbons, that is the reaction between the first carbon and the 5 carbon takes place, and thus, my dear students, the first carbon has got an extra bond, that is the bond between the first carbon and the fifth carbon, that is the linkage via oxygen. So this oxygen get another bond formed, and thus the first carbon and the fifth carbon get linked with the oxygen, and here you get the linkage bond between the first carbon and the last carbon, and the hydrogen which was present in and this first carbon has got an extra OH bonded to it. So this OH group which was there, instead of this, the reaction between first carbon and fifth carbon takes place, and this oxygen combines with this hydrogen combines with this oxygen, which gives an extra OH on the first carbon. And thus, my dear students, we have the cyclic structures of glucose. This cyclic structure have two possibilities. You can see these possibilities that on the first carbon, if we really study well, my dear students, that there are two possibilities for this cyclic structure of glucose. That on the first carbon, the OH group is present on the right hand side. This is the one possibility, and the OH group is present on the left side. This is the second possibility for the cyclic structure of glucose. Once again, let me revise, my dear students, that when the glucose is, when the first carbon of glucose combines with the fifth carbon of glucose, we get the cyclic structure of glucose. The cyclic structure of glucose can have two possibilities. First possibility is the OH group present on the first carbon is on the right hand side. The OH group present on the first carbon is on the left hand side, my dear students. If the OH group present on the first carbon is right hand side, we call it alpha D glucose, and if the OH group present on the first carbon is left hand side, we call it beta D glucose. And this is how, my dear students, even we understand the alpha D glucose and beta D glucose into the cyclic structure of glucose. So, my dear students, these anomers of glucose can also be understood into the cyclic structure of glucose. It is believed that glucose has presence of all these three formed simultaneously, and there is an equilibrium formed among all these three structures. So all these three structures that you can see here, one is open end structure. This is the structure number 1. This is the structure number 2, which is the alpha form of glucose, and this is the structure number 3, which is the beta form of glucose. All these three structures are found at equilibrium with each other in glucose. The equilibrium is represented as this. You can see here that there is always an equilibrium established among these various forms of glucose. This is the open chain structures. This is the alpha plus D glucose. This is the beta D glucose. In beta D glucose, OH is present on the left side. In alpha D glucose, the OH is present on the first carbon right side. And this is the open structure of glucose. And this is how, my dear students, we can understand about the these various forms of glucose which are in equilibrium with each other. My dear students, here I have something to you please note it down that glucose has plus 52.5 degree rotation, fructose has got minus 92.4 degree rotation, and this minus sign represents the levorotatory structures, my dear students. Please note it down somewhere in your notebook so that you can understand the concept whenever it is needed. Alpha D plus glucose, beta D plus glucose, both are called the anomers of glucose, and they are in the cyclic forms represented by a Haworth projection. So this type of representation of glucose is known as the Haworth projection, and here this Haworth projection in pyranose ring from where 5 carbon with 1 oxygen are bonded in the hexagonal form. So this glucose, as it has got the pyranose ring, this kind of compound of carbon, my dear student, is known as pyranose, and that is why this glucose in the alpha D form or beta D form is also known as the glucopyranose. We call it as a glucopyranose, and this structure of carbon is known as the pyranose structure, and that is why this is glucopyranose. You can see that this is the carbon number 1, this is the carbon number 2, carbon number 3, carbon number 4, carbon number 5, and this is the carbon number 6. Now you can easily understand, my dear students, that on the first carbon the OH is present on the OH is present on the first carbon, OH is present on the right hand side. You can see here. So this is the right hand side, which is the lower side. And here, if you look into the beta D glucose, this is 1 number carbon, second, third number, fourth number. This is the 5th number carbon, and this is the 6th number carbon. You can see on the first carbon, this OH is present on the left hand side. So this is left hand side, this is the right hand side, and that is how you can identify the alpha D glucose and beta D glucose, which is nothing but glucopyranose. Same way, my dear students, galactose have the same structure where we can call it as a, this is the galactose, this is what it is known as the beta galactose, and this is what it is known as the alpha galactose. Again, you can see that the right and left hand side, you can see that here when OH is present on the left hand side, we call it beta galactose, and OH is present on the right hand side, we call it alpha galactose. So alpha galactose, right hand side OH, beta, left hand side OH. Now you can easily remember it, very easily without any doubt. Let's move forward and study about the fructose, which is the another sugar that we get it. Glucose is called glucopyranose because alpha D glucose is alpha D plus glucopyranose, and beta D glucose is also called beta D plus glucopyranose. Glucose is aldohexose, but fructose is aldoketose. So in fructose we have ketone group present as a functional group, and fructose is your ketohexose with following open chain structure. So now, my dear students, here we can see the open chain structure for fructose. This is the carbon number 1, 2, 3, 4, 5, and 6 carbon are also present in fructose. On the first carbon, my dear students, you can easily say actually it is CH2OH group present on the first carbon. Same way, on the sixth carbon also we have CH2OH group present. On the sixth carbon also we have CH2OH group present. On the second carbon we have a carbonyl group. On the third carbon, OH is on the left hand side and H is on the right side. But on both the fourth and fifth carbon we have OH on the right side. So we can easily understand the open chain structure as first CH2OH, then COO, then OHH, then HOH, HOH, and CH2OH. Same way, this is the fifth carbon, which is chiral. And on the basis of that, we, as the OH is on the right side, my dear students, we call it as a D-fructose, capital D-fructose, because the OH group on the highest chiral carbon is on the right side, and that is why we call it D-fructose. This D-fructose is also in the equilibrium with the alpha D-fructose because there is a reaction takes place between, this is the first carbon, this is the second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon. So in fructose also there is a reaction takes place between the second carbon and fifth carbon, and it turns out to be a cyclic structures, and thus we have this alpha D fructose where the OH group is present on the right hand side. So we call it alpha D group, alpha D fructose. When this OH group is present on the left hand side and CH2OH group present on the right hand side, we call it beta D glucose. So even beta D fructose, so even fructose has a anomers, and these anomers of fructose are called alpha D minus fructose and beta D minus fructose. Remember, fructose rotates the plane of polarized light into the anti-clockwise manner, and that is why fructose has got the negative sign, or fructose is levorotatory in nature. Fructose is formed by the levo-rotation, or fructose has a levo-rotation, and that is why fructose is called D minus fructose, alpha D minus fructofuranose, and alpha D minus fructofuranose. Now in fructose we have the 5 carbon ring, and that is why this kind of ring of carbon is known as furanose. So that is why, my dear students, we call it, we call fructose as a fructofuranose. For alpha D fructose, we call it alpha D minus fructofuranose, or we call it, for beta D fructose, we call it beta D fructofuranose. Here you can see that this is what it is known as the pyran ring. This is what it is known as the furan ring. You can see the similar kind of ring into the fructose, and that is why we call it as a furanose. So you can see that in the furanose ring, this is the carbon number 1, this is carbon number 2, this is carbon number 3, this is carbon number 4, and this is carbon number Sorry, I made a mistake, my dear students. Uh, Let us give number. This is carbon number 1, this is carbon number 2, this is carbon number 3, this is carbon number 4, 5 and 6. You can see that between the carbon number 2 and carbon number 5, we have the oxygen present in between these two. And you can see when OH group is present on the right side, we call it alpha D fructose, and when OH group is present on the left side, we call it beta D fructose. This is how we understand the Haber projection for fructose, and the various anomers of fructose. They are the alpha D fructose and beta D fructose. Now, my dear students, we want to study about the disaccharides. The first part of disaccharide is sucrose. On hydrolysis of sucrose, it is given alpha D fructose and beta D fructose. You can see that we have here the alpha D fructose where OH group is present on the right hand side, and beta D fructose where the OH group is present on the right hand side. This is how from sucrose we get alpha D glucose and beta D fructose. If you look at the structure of the sucrose, we can find that this alpha D glucose and beta D sucrose are bonded with the carbon number 1 to of the alpha D glucose to the carbon number 2 of the beta D fructose, and that is why this linkage is known as the C1C2 linkage. It is known as the C1C2 glycosidic linkage between the alpha D glucose and beta D glucose. So we can easily say that when sucrose is formed, the structure are inverted of glucose and fructose, and that is why it is known as the process of inversion of sucrose. Please remember that this structure of glucose is completely inverted, and that is why you can see that this H is represented on the upper side, whereas this CH2OH is represented at the lower side of the fructose. So when glucose and fructose combines to make sucrose, the structure of fructose get inverted, and that is why the process of preparation of glucose and fructose from sucrose is known as inversion of sucrose. Actually, sucrose is dextrorotatory, and due to the inversion, the alpha D glucose formed, which is having the rotation plus 52.5 degree, and we get beta D fructose, which is having the total rotation minus 2.4 degree, and due to the mixture of these two form, overall rotation turns out to be minus in sucrose. So when hydrolyzed dextrorotatory sucrose, sucrose actually it is dextrorotatory, that is plus sign, but when it is undergoing the hydrolysis, we get levorotatory product, because the rotation of fructose and alpha D glucose, if we mix them, or if there is a mixture, overall rotation becomes minus. So sucrose, plus sucrose is converted into minus overall mixture of alpha D glucose and beta D glucose and beta D fructose, and that is why this is even one of the reasons that why this process of formation, the process of formation of alpha D glucose and beta D glucose from sucrose, this process is also known as the inversion. So this is how, my dear students, we consider the process of formation of, thus conversion of sign of plus to minus or dextro to levo due to the inversion of the glucose. This kind of process is known as the inversion of sugar or inversion of sucrose. We have maltose, my dear students. Maltose, when undergoes the process of hydrolysis in the presence of the enzyme maltase, it gives us the two molecules of alpha D glucose. Here in the two molecules of alpha D glucose, if you look into the structure of maltose, we have C1C4 glycosidic linkage obtained between this alpha D glucose and alpha D glucose. Here in maltose we have C1C4 glycosidic linkage. You can see that this is the first, second, third, fourth, fifth, and sixth carbon of the one glucose, whereas this is the first carbon, second carbon, third carbon, fourth carbon, fifth carbon, and sixth carbon of the another glucose. So they are bonded C1C4, and in maltose we have C1C4 glycosidic linkage found. Now, my dear students, we want to study about the lactose. So when lactose undergoes the hydrolysis, it gives us the galactose and glucose. The galactose that we get is beta D galactose, and the glucose that we get is beta D glucose. So my dear students, you can see that between the beta D galactose and beta D glucose, we have a C1C4 glycosidic linkage, and this is how you can see that beta D galactose is the this is the structure for our beta D galactose, my dear students, where the OH group is present on the upper side, and same way there is an inversion of the glucose where the OH group which was present on the glucose is utilized to make the C1C4 glycosidic linkage, and this is how the galactose has been formed. Galactose is having C1C4 glycosidic linkage, and which is a reducing sugar in nature. Now, my dear students, we want to study about the polysaccharides. So let us study about the polysaccharides. Polysaccharides contains the polysaccharides contain a large number of monosaccharide units. Please remember that these polysaccharides contain the large number of monosaccharide units, and that is why they are called polysaccharides. Once again, polysaccharide contains a large number of monosaccharide units joined together by the glycosidic linkage. These polysaccharides are the most commonly uh, encountered the carbohydrates in nature. So in nature we get most of the polysaccharides. They mainly act as a food storage or structural material for the various living organisms. So these polysaccharides are either the food storage material or the structural material. We know that many of the part of the plants are made of cellulose, which is a polysaccharide. First, we want to study about the starch. Starch is the polymer of alpha D plus glucose, and it has two types of structures. So starch is the polymer of alpha D plus glucose, and starch has two types of structure. First structure is called linear structure, another structure is called branch structure. Let us name the first structure, which is linear structure. Linear structure is known as the amylose, and the branch structure of starch is known as amylose pectin. Linear structure of starch is water soluble, whereas branch structure of starch is water insoluble. Linear structure of starch is a component having 200 to 1000 monomers of alpha D plus glucose and forms 15 to 20% of the starch, and it has C1C4 glycosidic linkage. So please remember the linear structure, which is known as amylose, which is water soluble. It contains 15 to 20% part of our starch, and it has 200 to 1000 monomers of alpha D glucose, and it has C1C4 glycosidic linkage. Now, my dear students, we want to study about the branched part of the starch, which is called the amylopectin. This amylopectin is water insoluble. It contains almost 80 to 85% of the starch, and it is formed by this component. It has a linear as well as the branch structures, and as usual the linear part has got the C1C4 glycosidic linkage, but the branched part has got C1C6 glycosidic linkage of the monomers alpha D plus glucose. This we can easily understand from the figure of the structures. My dear students, we have the linear part, which is known as amylose. We have branched part, which is known as amylopectin. Here you can see that there is a simple line of the alpha D plus glucose, up, whereas here you have simple line as well as the branches also you can see here easily. So my dear students, the amylose has got the simple linear, and this is C1, this is C4, and it has got the C1, C4 glycosidic linkage. But here the amylopectin has got the C1 and C4 linkage for the linear part, which is the linear part, but when it comes to the branch part, this is C1 of this glucose structure, and this is the C6 of the another. So branch part has got C1, C6 glycosidic linkage, whereas the linear part has got C1, C4 glycosidic linkage, whereas in amylose only C1, C4 glycosidic linkage is formed. This is water soluble, whereas this part is water insoluble. This is water insoluble. This makes only the 15 to 20% part, my dear students. This is water insoluble. This makes 15 to 20% part, whereas this part makes 80 to 85%. So this is how, my dear students, we can easily study about the linear as well as the branch part of our starch, which is known as the amylase or amylopectin. Now, my dear students, we want to study about the cellulose. It is the bodybuilding material in the plant. So most of the plants, their body is made up of the cellulose. It makes the part of the plant which are supporting or preparing the outer part of the cell. We know that cell wall of each plant cell is made up of cellulose or amylopectin. So cellulose is the polymer of beta D glucose, in which beta D glucose units are joined by the C1C4 beta linkage. Please remember that here the linkage is very different. This kind of linkage between the glucose unit, you can see that this is the linkage part. The C1 of the this beta D glucose and C4 of the another beta D glucose, they are bonded in this manner, and this is what it is known as the C1 C4 beta linkage. So it is beta glucose glucosidic linkage, where we have the monomer beta D glucose. We have seen that in starch we have alpha D glucose as a monomer, but in cellulose we have the beta D glucose as a monomer. The linkage is here also C1 C4 glycosidic linkage. Now, my dear students, we want to understand about the another poly polysaccharide, which is known as the glycogen. The carbohydrate stored in the body in the form of the glycogen. So in our body, my dear students, all the carbohydrates are stored in the form of the glycogen. So glycogen is called an animal starch. Glycogen is also called an animal starch. This is the type of material that has structure similar to the amylopectin. So glycogen has very similar structures like the amylopectin. That means it has got the linear structure, it has got the branch structure. Sorry, the linear structure has got the C1, C4 glycosidic linkage, as well as the branch structure has got the C1, C6 glycosidic linkage. The same is found into the glycogen, but glycogen is more branched than the amylopectin. So the branching that are found into the glycogen is more than the branching found into the amylopectin. Glycogen is more branched and present in the brain, liver, and muscle of living organisms. So glycogen is present in the brain, my dear students. It is present in the liver. Glycogen is present into the muscle. So that whenever needed, glycogen can be converted to glucose to produce required amount of energy. So when this glucose is stored in the form of glycogen in the brain, in the liver, and in the muscle, and whenever the energy is needed, this glycogen is converted into glucose and the energy can be produced. Glycogen is also found in the yeast and fungi. Please remember, my dear students, that glycogen can also be found into the yeast and fungi. Now, importance of the carbohydrates. So, what are the important points about the carbohydrates that we need to study here in this part of the chapter? Let us study about the various important applications or various important points about the carbohydrates. Let us study, my dear students. Carbohydrates are essential for the life of both plants and animals. They forms a major portion of our food. So carbohydrates are the major portion of our food and very essential for our life. Honey is used as an uh, in, uh, source of energy by the weights in an Ayurvedic system of medicines. Honey, which is made up of dextrose or glucose, which is an important source of energy, and this important source of energy has been utilized for ancient time, and our Veds, they use or they are the Ayurvedic acharyas, they have used honey as a source of energy with the various medicines. Carbohydrates are used as a strong molecules as starch in the plants and the glycogen in the animals. So these carbohydrates are used as a strong molecules, and they are used in our body or body of the animals and plants. Cell wall of plant and bacteria is made up of cellulose. So cell walls are made up of cellulose, which are carbohydrates. We have furnitures in our home. So we have furnitures and any other materials from our home which are made up of wood, and it is the form of cellulose. So all type of woods are the form of the cellulose, and this is how, my dear students, we understand that wood is nothing but your cellulose. So all woods, all type of woods are made up of cellulose, and cellulose is a starch. We have the clothes made from the cotton fibers. These cotton fibers are also cellulose. Cotton fiber material and many other important industrial material and textile. We have papers, we have liquors, and we have breweries. These all contains the various types of carbohydrates in them. Same way, my dear students, the genetic code that is in the form of the either DNA or that is in the form of the RNA. These all genetic codes, uh, which are DNA or RNA. They contain the 2-aldopentose. That means they even contain sugar with them, which is a carbohydrate. The name of the sugar is either D-ribose, which is in the RNA, and 2-deoxy-D-ribose, which is present in the DNA, and this is how they are very important for preparation, or carbohydrates are very important for the preparation of DNA and RNA. Carbohydrates found in carbohydrates found in the biosystem in combination with the many proteins and lipids, my dear students. So carbohydrates are also found in the biosystem. They are they are combined with the proteins and they are combined with the lipids. So these were the some of the important applications, or some of the important points about the utilization or importance of carbohydrates. I hope, my dear students, you are enjoying your studies, and I wish you happy learning. Thank you. And if you have any doubts, you can ask me the questions.